Hello, all you cool cats and kittens. This is Carol Baskin from Big Cat Rescue, and we're going to talk about guinea pigs. So, have we heard of guinea pigs? I'm sure we have. Let's keep talking about this model of genetics that we've been learning about, which is called complete dominance. This is just one way that things can get inherited, from one human to another. And in this complete dominance thing, this is all of the stuff that we've been talking about. And in complete dominance, we say that there is maybe only two alleles for a gene, and that one is dominant and one is recessive. And we also say that as long as there's one dominant allele, that's the characteristic that will show or be expressed. So at least one dominant allele equals that trait is expressed. And so, if you are big H, little h, you are heterozygous, but the only characteristic that you see is whatever that big H1 is. In order to express that recessive allele, you need to be little h, little h. Now, there's this kind of perception out there in the world that, all good characteristics are dominant and all bad characteristics are recessive, but that's not always true. Just a couple of examples. First of all, the gene for dwarfism is actually because of a dominant allele. Now remember that there's a difference between different kinds of dwarfism, so not all individuals that you see that are dwarves or little people are what we're talking about here. So dwarfism is governed by a dominant allele. And a lot of you might think, oh, well, being a dwarf is not great, and so therefore it's recessive, but that's not true. It's not true in all cases. You all are pentadactyl, which means that you have 5 digits on your hands. Penta meaning 5. Being pentadactyl is actually a recessive allele. There's plenty of people born all the time with a 6th digit coming off of their thumb, and that is removed quite frequently. Normally, every year in my classes, I have somebody who had that 6th digit surgically removed when they were younger. Actually being more than pentadactyl is a dominant allele as well. Another good example of this, and this is one that you must know. This is one that you must know. This is an IB requirement. Has to do with a disease called Huntington's disease. Huntington's disease is a little bit like Parkinson's disease. This person will have a lot of muscle spasms. They'll have dementia, and all this is because their neurons are degrading. So it has to do with a gene on chromosome 4, and this gene codes for a protein that is called huntingtin. So we've got our protein huntingtin, and this is a real interesting gene. This gene has a bunch of CAG repeats, anywhere from like 10 to 25 of them. And there's this odd problem where if you have more than 28 of them, they start to copy themselves, so there are more of these. And once you get up to 40, or more, that protein misfolds. And when it misfolds, it causes your nerves to degenerate. So you might be thinking, okay, well, if this is true, then how come it is that this still exists in our world? This doesn't make any sense. If it causes this neurodegenerative disease, how can this be in our gene pool? And the answer to that is it doesn't show until you're about 40, 50 years old. And by that time, you probably already had kids, at which point you pass that gene on. Maybe or maybe not, depending on what your genotypes are. So, is this a dominant or a recessive characteristic? The answer is, this is a dominant characteristic. So, if you are big H, little h, you're heterozygous, but because of this dominant big H right here, you will have Huntington's disease later on in your life. Now, let's imagine that this person mates with and has children with somebody who is recessive for this characteristic, which means they are not going to get Huntington's disease. All right, let's do a real simple, quick little Punnett square here. We'll separate the alleles to show our gametes here. We've got big H, little h, big H, little h, little h, little h, and little h, little h. There's a 50% chance that your child will end up getting Huntington's disease if this is the case. So, that's one disease. There is a second disease related to this complete dominant style of genetics that you need to know. And that disease is one that you're already pretty familiar with, and that is cystic fibrosis. Just a recap. Cystic fibrosis has to deal with a problem with the protein CFTR. And CFTR moves chlorine ions in and out of your cells. And if it doesn't work, the chlorines get all trapped up in your cell. So you get a whole bunch of chlorines here. And our cells are covered with these layers of mucuses, especially in all of our cells that are that make up the tubes. Since stuff is going to pass through those tubes, there needs to be a layer of mucus to protect the cells. But if there's too much chlorine in here, in these cells right here, then that means there's a low concentration of water in there, and water then will want to move into the cells. That makes this mucus super thick and hard for stuff to cross, which is why these people can't get digestive enzymes from their pancreas to their intestines. They have a real tough time breathing because they've got this thick mucus built up in their lungs. It's hard for the gases to get through that. And these people are normally infertile too, because all of our sperm and eggs and things like that travel through tubes, and if those tubes are all blocked up with thick mucus, it's hard to get through. Now, this disease is inherited just a little bit differently. It does follow our complete dominance model, which means that there is a dominant and a recessive allele. But unlike Huntington's disease, which was caused by a dominant allele, cystic fibrosis is caused by a recessive allele. A recessive allele. So, if you, let's say you were heterozygous for cystic fibrosis, big F, little f, and you were to mate with another person who was also big F, little f. Neither of you have the disease because you need to be completely recessive. And if the dominant characteristic is that you don't have cystic fibrosis, then neither of you would know this unless you went and got tested genetically. So, in big F here, we've got big F, little f, big F, little f, and we have little f. Right here, this person right here. If two heterozygous people were to have children, there is a 25% chance that their children have cystic fibrosis. This person wouldn't have a very amazing life, but they could still do a whole bunch of stuff, run out of breath real easily, not have a lot of energy, be restricted in a lot of activities, not have their bacteria like to build up in the mucus, so they kind of have to do what we're doing right now and live a life of isolation a little bit because you can't be around a lot of people. Not great. And so there is a career called genetics counseling where you can very easily do Punnett squares for people based on their family histories and some simple DNA tests, and you can tell them what kinds of diseases they may have and may be likely to transmit. That brings us up to these guys up here. These are guinea pigs. This guy right here is my spirit animal. Looks like I do when I wake up a lot of times. So guinea pigs, we're going to talk about guinea pigs really quick for a second because we as human beings are not just one trait people. We can look at ourselves and we can predict what our children might be based off of one trait. But that's not terribly interesting. And a lot of times the important things are combinations of traits. So how can we do the kind of genetics we've been doing, but do probabilities when there's more than one gene involved? It's a pretty simple process. And we're going to talk about two characteristics for guinea pigs. We're going to say that guinea pigs can have two different hair colors. The dominant is black hair, big B, and the recessive is white hair, which is little b. Now, most guinea pigs are short-haired, and we're going to say that is big H. These are short-haired guinea pigs, and little h are long-haired guinea pigs. Okay? Now, let's imagine that we have two heterozygous individuals. A guinea pig that has black hair, heterozygous, and also short hair, heterozygous. And we have another individual that is the same kind of thing. Well, this kind of Punnett square is real challenging, and it's something that we would do in HL. Conceptually, it's hard for some people to visualize, so we don't even do it in SL. This is an HL thing. So let's do two Punnett squares. One where we just combine our B's, and one where we just combine our H's. Pause your video, do these two Punnett squares really quick, and come back. Okay. So let's see, if these are our two guinea pigs that mate, what will happen? What are our odds of getting some of these combinations? Like, for example, black, long hair. What if that's something we're going for? What if we're guinea pig breeders and we really want to make sure that, we are seeing the kind that we want? So it's real easy. What we do is we take the individual probabilities. So the odds of getting a black-haired guinea pig out of this cross are 3/4. And the odds of getting a long-haired guinea pig out of this cross are 1/4. And the rule in probability is that you multiply these probabilities together. When you multiply fractions, you just multiply the top, which is 3 times 1 for 3. And the bottom, 4 times 4, is 16. So there's a 3 in 16 chance that the guinea pigs will come out black, long-haired. What about long, white hair? These odds are a 1/4 and a 1/4 for 1/16. So not good odds on getting a white, long-haired guinea pig. But the good news about that is, since white, long-haired guinea pigs are little b, little h, if you were to get one of them and mate it with another one, all of your guinea pigs would be little b, little h, if that's what you were going for. What about getting black short hair? These are both the most common. Black short hair, and that's going to be 3/4 times 3/4, and that's going to be 9/16. Now you'll know you did it right if you do all the possible combinations. What's our other possible combination? And that is white short hair. You'll know you're doing this right if, when you do all of the possible combinations, whoops, that's not 9, that should be a 3/16. They add up to 16 out of 16. So 3/16 plus 1/16 is 4/16 plus 3, another 3 is 7/16. Plus another 9 is 16/16. So we did that right. All right. But enough about that. Let's go into our last part of today's notes. And this is real short, real easy. You should be totally cool with this one. Okay. Our last part of today's notes has to do with these two right here. These are our sex chromosomes. And you know there's two kinds of sex chromosomes. So these are chromosomes, which means they have little pieces of information on them that are that contain, information for a trait. So we've got our genes here, And, these chromosomes can be passed on, down to your kids and things like that. But we have a real interesting situation here, because if we have a gene that's on the X chromosome right here, whether you have a boy or girl, it's going to be on that X chromosome. However, we've got this issue, and the issue is that guys don't have two X chromosomes. And so if there's a gene on the X chromosome, then the trait, the characteristic that is going to express in the male is only dependent on whether it's there in one spot. And so in this case, you're not heterozygous or homozygous if you're a male. You're what we call hemizygous. It's not important to know, but just, those cool vocab words are awesome. And so let's take a look at two particular characteristics that IB expects you to know, and that are really interesting as far as genes that are on your sex chromosomes. So characteristic one is your ability to see color. This is governed by a single gene. If you have the dominant version of that gene, you can see color. But if you have the recessive version of that gene, you are some degree of colorblind. Not all colorblind people see grays, and that's it. Some colorblind people just have trouble with certain shades of things. I'm one of those people. So if we were to do this in class, as soon as I said that, a bunch of you would be like, "What color's my shirt?" And I'd be like, "Go away." But no, red's all look the same and things like that. Now, because these are on the sex chromosomes, and because males only have one of these two alleles, whereas all females have some combination of two of these, we write these down a little bit differently. So how we write these alleles down is by using an X or a Y for whichever chromosome that we're talking about right there. And then as a superscript, like a power, we include what the allele is. And we still use capital and lowercase to show dominant and recessive alleles. So this is a male that can see color. This is a male that is colorblind right here. Females, same thing, except there's no Y that we're going to write here, just X's. This is a female that can see color. And this is also a female that can see color. Because she has a dominant allele, that's the only thing we see. We do call her a carrier for color blindness, but it doesn't express, and she can see color fine. So what would a colorblind female's genotype look like? And the answer is X little b, X little b. We do these Punnett squares the same exact as other people. We do these Punnett squares the same as we do all of our other things. So let's take a female that is a carrier for color blindness, and let's have this female have children with a male who is not colorblind. So let's set up our Punnett square by separating our alleles out to show that only one allele passes from the male and one allele passes from the female. Okay, so this child is X big B, X big B. If they had this child, this would be a non-colorblind female. This child would be X big B, X little b. This child would also be a non-colorblind female, but they would be carrying the colorblind allele. This X big B Y would be a male that is not colorblind, and this is a this would also be a male, but this is a colorblind male. This is me. This is how I became colorblind. All right. So colorblindness is pretty easy, but there's a much more interesting sex-linked trait. There's another interesting sex-linked trait because it's on our sex chromosomes. And that is a disorder called hemophilia. Now, when you think of hemophilia, you should probably think of blood. And that's because hemophilia is a disorder where your blood cannot clot. If it can, it's very difficult. And you might be like, oh, well, just don't get caught on the outside. Well, we bleed internally all the time. I mean, that's what aneurysms and bruises and things like and hemorrhages and things like that are. So this happens all the time, and if you can't stop that, then you could bleed out inside your own body or outside your own body if the cut is severe enough. This is also governed by a recessive allele. So big H is normal blood clotting. And little h results in a hemophiliac person. So, if a female carrier of hemophilia was to have a child with a male hemophiliac, what would our Punnett square for this cross look like? I tried to fix that, but that's not fixable. Okay, there we go. All right, so this is our female up here. All right. So this child would be a normal blood clotting female, but they would be carrying the hemophilia gene. This would be a hemophiliac female. This would be a male without hemophilia, normal blood clotting, and this would be a male with hemophilia. So if these individuals had children, there would be a 50% chance that their child would be a hemophilia. There's a really interesting story about hemophilia in royal families. When royal families back in the day used to inbreed a whole lot, you used to see these kind of characteristics pop out all the time. Because when individuals mate with each other, there's a better chance that these recessive characteristics come into play. So if you have a bunch of carriers of diseases that are heterozygous like this, that are mating with each other through lots of incest and interfamilial relationships and things like that, there's a greater chance that you end up with these individuals that have the disorder and the disease. It's that genetic variety that's really important with humanity. And this is one of the reasons why inbreeding and incest are really, really, really bad, because individuals that have poor recessive characteristics like this, there's a greater chance that those things come out. All right. Do the sex-linked trait worksheet on your notebook. Give yourself a colorblind test, and I'll send out a reading tomorrow.